So, Deepak, hi.
Hey, hi there, Tim.
Good. I think we're probably about ready to start. I won't say, I'll forgive you the briefest of introductions because everybody reads your bio before they come here, but I will just to say that you're an associate professor of petroleum engineering at the University of Oklahoma. And unconventional reservoir modeling and management and EOR are some of your interests with machine learning. And you're going to tell us about revealing the unobservable with molecular dynamics simulations. So Deepak, the floor is yours.
Absolutely, thank you. Let me go ahead and share my screen. I want to thank you all for the opportunity to present some of my work this evening to most of you and afternoon for me, because I'm across the pond in Oklahoma. Tim, thank you very much for extending an invitation to me to come and speak at the SPE London evening meeting. And so all of you can see my screen, right? I suppose so. Okay, good. So having said that, I'm going to be talking about something called molecular dynamics, which will help us see something that we can't really observe. And in fact, the things that I'm going to be talking about is the science of small things. What do I mean by the science of small things? Well, we have a schematic right up over here, which illustrates the point. We have what we're very, very familiar with right now. We have the COVID virus, which is roughly about 100 nanometers across. And I'm going to be talking about stuff that is really, 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 really small. We're talking about maybe a couple of nanometers across or maybe tens of nanometers across. So that's what I'm going to be talking about. And I'm a faculty member here at the University of Oklahoma. I wear two hats. One of them is where I do this kind of stuff, where I'm modeling fluid and fluid rock interactions. The other hat that I wear is related to machine learning. So I was very glad to sit in for Fatah's talk that we just had about over the past 2 hours. And with that, we'll get started over here. So this is the outline of my talk. I plan to keep it to about an hour and then open it up for questions. Since I'm a reservoir engineer by training, by background, most of the applications that I'm going to be talking about are reservoir engineering related. But how we're going to organize this talk is first talk about what exactly is molecular dynamics, then see what does it involve, and then look at some applications. So that's the whole idea over here. And so we'll begin with introduction to molecular dynamics. So why do we need to model really, really small things like atoms and molecules? Well, it turns out that we can't really observe lots of things with microscopy, even with some of the more advanced microscopes out there, scanning electron microscopes and scanning tunneling microscopes. We can't really observe these things. And again, the other thing is we can't observe them at high temperature, high pressure, or in their native environments. I'm talking about processes like photosynthesis. I'm talking about things like clay swelling. I'm talking about all of these non-equilibrium processes. And so if you want to begin to get some insights into what's happening in these processes, what most people do is they set up a molecular dynamics simulation setup. And as with most things in petroleum engineering, this is not something that we invented or something that we developed. We borrowed this from other sciences. And specifically, when we talk about molecular dynamics, we borrowed this specific technology from the medical field, as well as from chemical engineering. And just to show you how people in medicine or drug discovery use this, we know, of course, right now we're all super interested in the COVID-19, the SARS-CoV virus, because we've been living through this pandemic for the last year and a half. And of course, we now have vaccines that neutralize this virus to a certain extent. And the question is, well, how do we develop vaccines that would disable this virus? Now, of course, you could inject a whole bunch of people with this virus and then try a whole host of cocktails, drug cocktails, and see which one's more efficacious. Now, you know now that that's going to be dangerous. It's going to be time consuming. And it's probably going to be something where you probably may not have any outcomes at all just because you didn't choose the right cocktails. So, well, how do we do this? It turns out that we can do this by setting up a molecular model of the SARS-CoV virus. And you can see that it has a specific structure. The reason why it's called a coronavirus is because it has this corona out over here, and it has a certain molecular structure to it. It has a certain physical structure to it. And if you're going to be disabling something, whether it be a bacteria, whether it be a tumor cell, whether it be a virus, essentially you're harnessing something like a lock and key mechanism. You want something that would go and lock into the physical and chemical structure of this virus and disable it. And that's essentially how we're developing drugs. We're setting up these models and developing other models of drugs that would perhaps go in and disable these tumors and bacterial cells and viruses and things like that. So we do all of that in a simulation environment. So why is that advantageous? Because then if I know that certain drugs work in a simulation environment, then I can begin to start developing those drugs in a lab and then start testing them out on petri dishes of bacterial cells or tumor cells and things like that. So this is the first step towards drug development. This is how every single drug is pretty much developed. And of course, there are a whole host of other applications as well. Some of you may have already had your dinner. I have yet to have my lunch, but when we digest our food, we are essentially absorbing our food as glucose and amino acids and things like that. And the amino acids are not bioavailable. So in fact, if you want to actually use the amino acids need to link together, form a long chain. And even then, it doesn't form a protein. That protein needs to fold in amongst itself in a very orchestrated, choreographed fashion to form something that looks like this, that you can see on my screen out over here. And only then does it become bioavailable. And turns out that we can't observe this process in a microscope. The only way you can model some of these processes is using molecular dynamics. And why would we be interested in modeling the molecular structure of these proteins and how it comes together? The reason why we want to do that is because if these proteins don't fold in the right fashion, you end up with diseases like Alzheimer's and things like that. So if you want to develop a cure for Alzheimer's, you need to know why the protein's folding in a wrong fashion so that you can develop something that will allow the proteins to fold in the right fashion. So with that background, we'll jump into why specifically I'm talking about molecular dynamics in this talk and why you should be interested in molecular dynamics. Even though you may never use these tools, at least for the foreseeable future, but rather you would not use them directly, but you'd be using the results of some of these simulations. We're all familiar with this map. It's the map of the US, and some of you who work in the unconventionals have perhaps heard of some names like the Barnett Shale. You've perhaps heard of the Marcellus Shale up over here. Right now, something that's extremely hot is the Permian, which is down over here in West Texas and eastern New Mexico. Then, of course, we have the Bakken, which is in North Dakota and Wyoming, and then adjoining Canada. So extremely, extremely prolific shale oil, oil shale, shale oil and gas plays. And so prolific, in fact, that the US has been the largest oil producer for a very, very long time, probably close to about 8 to 9 years. I could be wrong on that, but definitely for a very, very long time. And it turns out that all of these are shales. And when we talk about shales, we're talking about really, really extremely tight reservoirs. So tight, in fact, that we're talking about nanometer sized pores. And if you're talking about nanometer sized pores, we still don't know how things flow through these nanometer sized pores. Yet we're producing these humongous, huge amounts of oil and gas from these reservoirs, but we still don't fully understand the processes that are driving the transport of these hydrocarbons through these extremely small pores. So to give you a sense of what size scale I'm talking about, over here, I have a length scale that's going from about centimeter to about 1/10 of a nanometer down here. And you have some man-made material on the right-hand side associated with these different length scales. We, of course, have carbon nanotubes, which have seen a whole host of applications. And then on the left-hand side, you have an ant, you have fly ash. You have fly ash down here. You have the human hair, which is about 50 microns or 100 microns wide. And coming all the way down, you have a DNA molecule, the DNA structure, which is about 2.5 nanometers wide. And then you have a methane molecule, slightly lesser than half a nanometer. Okay. Sorry, I thought somebody had interrupted me. Anyways, and then I want to draw your attention to this picture on the left-hand side over here. And what you're looking at is a scanning electron microscope picture of a shale. Specifically, this is a sample from the Barnett Shale. And this dark stuff that you see over here is what's called as Kerogen. It's the organic material present in the shales. It's the place where the hydrocarbons are hosted because it's hydrophobic. And this is where the hydrocarbons are hosted. You can see some pores which are extremely large, but you can also see some pores that are extremely small. That's what you're looking at. And just to give you a sense of why this matters, I want you to think about conventional reservoirs. If you're talking about conventional reservoirs, we're talking about pores that are about 50 to 100 microns wide in diameter. And we're talking about hydrocarbons that are roughly about 0.5 nanometer in diameter. So I can fill about 3 to 4 million methane molecules in a pore that's that large. Now, on the other hand, if I'm talking about a pore that's about only 6 nanometers across, then think about it. I can only fit 12 methane molecules end to end in that extremely small pore. And if you're talking about 12 methane molecules, you can't even talk about a phase. Is it oil? Is it gas? What is it? How do you even begin to talk about PVT behavior? How do you even begin to talk about what the molecules are doing? And it turns out that in those really, really small spaces, the influence of the pore walls themselves plays a huge role in driving how the fluid particles, how the fluid molecules move, how they're stored, and things like that. And so the only way we can observe this or model or say something about this is through molecular dynamics, because we're talking about extremely, extremely small stuff. Good. So what exactly is molecular dynamics? It's a very, very time-consuming, computationally expensive process. But in reality, what we're doing is we're constructing molecular models of various things that we want to study. So if you want to study the behavior of methane enclosed in a graphite pore or a silica pore, you will have molecular models of methane. You will have a molecular model of silica, and you put them both together, and then you try to understand how they behave at specific temperatures and pressures, so that you can say something about adsorption. Perhaps you can talk something about maybe coagulation or colloids or emulsions. You can quantify densities. You can say something about the volume of the material and things like that. So if you're going to be doing something like that, then what do we mean by or so how exactly are we going to be quantifying these phenomena, things like adsorption, things like density and volume and things like that? So it turns out that the process is rather quite simple. So I'd like for you folks to imagine a box that contains atoms of various sizes and different types. You have some red atoms, you have some green atoms, and you have some blue atoms out over here. Now, of course, we all know from high school chemistry that either there are going to be some forces associated with these atoms. They can be atoms or they can be molecules. It's either van der Waals forces or it's Coulombic forces. And so if I were to just show you what would happen if I run this simulation, unfortunately it doesn't, yeah. So if I run this simulation, then of course you can see the balls are just moving around, the atoms are moving around, and they're moving around because one specific, this atom over here, which is where I'm pointing to, is being acted upon by the forces exerted upon it by the surrounding atoms. And that's going to move the atom in a certain direction, in a certain velocity. And likewise for every one of the atoms or every one of the molecules in this picture. So again, it's nothing complex. This is just Newton's laws of motion. The only difference is that the forces we're going to be talking about are Coulombic forces. They're going to be van der Waals forces. So that's essentially what we need to keep in mind. And so you can begin to start playing games like this and see how the molecules move and whether they coagulate, what kinds of densities we're talking about, and things like that. Something to keep in mind is that since we're modeling the motion of each and every atom or each and every molecule, we're restricted in the kinds of systems that we can model. Typically, we're talking about 10,000 to about 1 million atoms. And the time scale of investigation is also not very long. We're not talking years or even days or even hours. We're talking about nanoseconds. So we're talking about something that's extremely, extremely fast, something that's extremely a non-equilibrium process. Okay, so with that background, what kinds of forces do we have? You can have bonded atomic forces. So of course, if you have a methane molecule or if you have water, then of course you have the bonds between hydrogen and oxygen or between carbon and hydrogen. You need to specify those bonds. And those bonds are of course not necessarily of a constant length. They can vibrate, they can twist and turn amongst themselves. And so you need to specify to the simulator that this is what these bonds can do. And fortunately for us, chemists have spent their entire lives telling us about these bonds. So we just extract those kinds of that information from that literature and then feed it into our model. Just like when you're doing a reservoir simulation model, you need to specify what kinds of fluids you have and what kinds of relative permeabilities and porosities and permeabilities you have. Here, the model inputs are going to be the types of atoms, the types of molecules, whether they're bonded and if they're not bonded. And if they're not bonded, of course, the only ways they're going to interact are van der Waals interactions and Coulombic forces. So we set up the simulation box and then we hit the run button and then wait for it to reach equilibrium, and then we analyze the results. So having said that, let me show you an example application of molecular dynamics in petroleum engineering. Why would you want to use molecular dynamics in petroleum engineering? I'll show you something right now. Okay, so on this specific screen over here, you're looking at different molecular models of different hydrocarbons. Here's a model of methane. Here's a model of ethane and propane. And of course, this is not a ball and stick model. When you represent this in a simulator, you would say that there's carbon, there's hydrogen, there's the bond angle, there's the bond length, and so on and so forth. But pictorially, this is how you would depict each one of these hydrocarbons, and nothing new. Okay, so we know butane, we know octane, we know the dimethyl naphthalene and asphaltene and resin fractions and things like that. And of course, since I'm talking to you about unconventional shales, really, really small pores, we're also going to talk a little bit about kerogen, which is that organic material that I was showing you a few slides earlier. So with this organic material, you're looking at one of the pieces that makes up that organic material. And it has a formula of carbon 242, hydrogen 219, and so on and so forth. This is what it looks like. If you were to look at this if you're able to look at this under a microscope, this is what it would look like. Each one of these dots represents a carbon atom. This red dot over here represents an oxygen atom. This blue one over here represents nitrogen, and the yellow one represents sulfur. So this is the molecular structure of one of the monomers that makes up kerogen. And of course, these are the molecular models of all of the hydrocarbons. So first things first, we need to construct a model. So how do we do that? We take, for instance, I'm going to take 16 monomers of kerogen, those big massive structures that you saw on the previous slide. I'm going to take about 160 methane molecules, and you can see the number of other hydrocarbons that I'm going to be taking. And I'm going to put them in a simulation box. So of course, I'm doing all of this virtually. I put them in a simulation box that's about 10 nanometers across. And since I'm just putting them together, this is of course not an equilibrium configuration. What I need to do now is I need to virtually apply some pressure. and increase the temperature and keep compressing the simulation box virtually so that things bend and the molecules rearrange themselves, and then finally reach a configuration which is an energy minima. And of course, this is a complex process. From this start to the model I'll show you on the next slide can take up to about 2 months, okay? And the scale, again, I want to emphasize over here, 10 nanometers across, not large by any means. But you're looking at all of the carbon dots, you're looking at all of the oxygen dots, there's some nitrogen over here, there's sulfur, and some of them are bonded, some of them are not bonded. So you have all of these things put into a simulation box, and then I go through that annealing process. I apply some pressure and temperature and slowly bring them all together so that they reach an energy minima. And so this is an energy minima. It's a square cube, roughly about 5 nanometers across on each side. And what you're looking at over here are the final pressure and temperature conditions of about 4,400 PSI and 180 degree Fahrenheit. You're again looking at the balls in gray, which are the kerogen atoms, carbon atoms. Then you're looking at some of the blue ones out over here. These are the intermediate species, being things like hexane and octane and butane and things like that. Then you're looking at some of the orange stuff over here, these orange balls, these are associated with methane. And you're also looking at some of the heavy and aromatic species in the slightly lighter, darker yellow over here. There are some water molecules too, and things like that. And so that's the final structure. And it's not quite apparent from this picture, where are the pores and where are the fluids located, why are the fluids restricted out over here, and things like that. So I'm going to show you a video show you a few other snapshots of this model. So first things first, I'm going to remove all of the carbon atoms associated with kerogen. And now you can just see the kerogen skeleton, and you can see these gray surfaces. And these gray surfaces are what we have rendered as the internal pore surfaces within this kerogen. And within these pores, you can see the fluid molecules residing. You can see these fluid molecules residing out over here. And then furthermore, I can remove all the fluid molecules as well. And then you can see the kerogen structure a little bit better. You can actually see a big pore throat out over here. And we can, of course, turn this around and play games and look at it from the top and everything else. But that's your kerogen model. And in fact, to see it better, I'm going to show you this thing as a part of a video. So we started off with this kerogen model with the fluids sitting in the pore spaces and these gray surfaces being the pore surfaces. So, and we're going to turn this around, and you can see that the fluid molecules are residing in the pore surfaces, inside the pores. Again, fluids residing inside the pores. And now I've removed all of the kerogen atoms, and you're just looking at the pore surfaces and you're looking at the fluid molecules. And if I were to pause this right here, you can see the fluid molecules residing in the pores over here. And this gray surface again encloses the pore volume within that kerogen blob. And now I've made the pore surfaces translucent, and you can see all of the fluid molecules contained within this kerogen model. Good. So all of that's fine, but what can I use this model for? So it turns out that you can see that I have some methane molecules over here. I have some of the other hydrocarbon species down here. I have some carbon dioxide over here. And so the next question I want to ask myself is, where are these different fluid molecules or these different hydrocarbon species? Where do they reside inside this pore model? And the reason why I want to ask that question is I want to know which hydrocarbon species are adsorbed onto the pore surface, which ones form a free phase. So if I did something like that, and then just draw the location of each one of the hydrocarbon species, you would find that, again, this is the same 3D cube with the pore surfaces. You can see methane molecules all over the place, occupying most of the pore volume. And the methane, it turns out that we can actually observe the methane moving rather freely. It's what we call as the free phase. It's not adsorbed onto a surface. Same thing with carbon dioxide. But on the other hand, if you're beginning to talk about some of the heavier molecules, things like the asphaltene and the resin fraction, or some of the heavier hydrocarbon species, and even some of the intermediate hydrocarbon species, things like octane and things like that, they tend to be, at these pressure and temperature conditions, adsorbed onto the surface. And so the question then becomes, okay, now I know this stuff. I know that some of the hydrocarbon species are adsorbed and some of them are free. What's special about that? The reason why this is special or why this is unique is because we are used to thinking about conventional reservoirs. When we think about a pore, we think about the entire pore being filled up with a fluid whose composition is uniform wherever you look at inside the pore. So I can go to another location within the reservoir as long as I'm not changing, going across a fault or something like that. The fluid composition is pretty much the same. But here the thing is that adjacent to the pore walls, I have heavy species. Closer to the centers of the pores, I have some of the lighter species, things like methane and propane and ethane and things like that. So I want to ask myself, why is this important? It turns out that this is important because if you were to be looking at the asphaltene and resin fraction, for instance, you can see some of these molecules blocking this pore throat over here. So if it's blocking this pore throat, it means that it's also blocking access for any solvent to move into Kerogen. If you were to imagine an enhanced oil recovery process, any solvent that wants to come in and mobilize some of the hydrocarbon, it's blocking access for that solvent. It's also blocking access for some of the molecules contained within the Kerogen to move out of that Kerogen and be produced. And now you can say, hey, this is a modeling construct. Can I believe you at all? It turns out that this modeling hasn't occurred in isolation. The reason why we did this modeling was because what we observed with lots of shale samples is precisely just what I'm showing you with this modeling diagram over here. So on the right-hand side, I have a picture of a native sample from the Eagleford. And a native sample means that it's a sample that was just collected and nothing was done to it. It just came out of the ground and we keep it over here, keep it on a desk and we image it. And you can see some pores over here. You can see the dark material, which is organic. And then, of course, if you clean this sample and then re-image it, you can very clearly see, going from the left to the right, I've opened up a whole bunch of pore spaces out over here, a whole bunch of pore spaces and a whole bunch of pore volume. And when we analyze the whatever we recovered after cleaning these samples, what we were recovering was pretty much this asphaltene and this resin fraction. So it turns out that this asphaltene and resin fraction, even when you bring it up to the surface, they're clogging all of these pore throats. They're clogging the pore throats even at high temperature and high pressure conditions. They're preventing fluids from moving. And we see that at other samples as well. And more importantly, what we see is that if you were to plot the porosity on the y-axis, for different shale samples, what you see is that if you take the native sample, you typically see a very, very low porosity. But then when you clean the sample and open up some of those pore throats, you are gaining access to more pore volumes and you actually measure a significantly higher porosity. It doesn't matter if this is belonging to the volatile oil window or the black oil window or the gas condensate window. That's something that we see all the time. So again, so molecular dynamics really was useful in telling us something about what we were observing from experiment. Okay, are there any other things that I need to be concerned about because some of the molecules are adsorbed and some of them are not? Some of them are free fluids. It turns out that whether the molecule is adsorbed or free also controls its mobility. Something that we've known for an extremely long time is the fact that even at pressures above the saturation pressure, if you're talking about an oil reservoir at pressures above the bubble point, or if you're talking about a gas reservoir at pressures above the dew point, in conventional reservoirs, we expect that if we're producing at pressures above the saturation pressure, the produced fluid composition should not change. And that's because there is no condensation, there is no evaporation in situ, in the subsurface. It's a single phase fluid that's moving through the pore spaces all the way to the wellbore and the surface, and we're collecting it over there. There should be no change in composition. Turns out that with the shales, we actually do observe that the compositions are changing, even if our producing bottom hole flowing pressures are above the saturation pressure. And that's extremely strange because that flies in the face of everything that we've learned about when we talk about fluid flow and maintaining things. Here's an example on the y-axis. There's time on the x-axis. And why do I say normalized composition? Because the composition at any point in time is normalized to the initial composition. So all the graphs start from a value of 1. And this black line over here represents the evolution of methane as a function of time. You have this green line over here, which represents the evolution of ethane as a function of time. And then, of course, you have propane, you have butane, and you have pentane, and so on and so forth. And what you're seeing is that, of course, initially, there's quite a bit of noise. We can't really see anything that's going on. And that's because when you're talking about shale wells, you have a whole bunch of cleanup, and you're producing your frac fluids and things like that. But after a certain period of time, you can very, very clearly recognize the signature of a moderate increase in methane fractions and a definite decrease in some of the heavier fractions. And this was, of course, for a relatively light gas field, but we also see this with oil fields and things like that. So if you're producing above the fluid dew point, why would you see something like this? That should not occur at all. At least for conventional reservoirs, we don't see something like this. So that's what we set out to investigate. And here's our molecular model of kerogen. Again, you can see the balls over here. You can see the dots representing kerogen. We have the asphaltene and resin fractions, some of the heavy and aromatic species, some of the intermediate species, and methane, of course. This is our pressure. This is our temperature. And of course, a slightly larger model, closer to 10 nanometers as opposed to 5 nanometers that I showed you previously. And this is the composition. My mouse is dead. This is the composition of the oil out here. Okay, so the oil sitting in the pore spaces, and you can see the oil sitting in the pore spaces all around over here. And then there's the kerogen blob. Okay, let's go to the next slide. And what we did was that we took that kerogen blob and we placed 2 kerogen blobs adjacent to each other, and we separated them out by a distance of about 13 nanometers. And why 13 nanometers and not 12? It was just convenient for us. We just chose 13. There's nothing sacrosanct about 13 nanometers, but we just separated them by a certain distance. And what we want to do now is start seeing what would happen if I were to start producing these hydrocarbons from within kerogen and allow them to move towards this void space out here. You can think of this void space as like a very, very small micro or nanofracture. And there are micro and nanofractures in shales. We've seen enough number of them on SEM pictures, things that are just a couple of nanometers across. So this is not out of the realm of reality whatsoever. So we just want to see what fluid molecules are going to be produced. Good. So that's our model. And here I'm going to be showing you a video of what exactly happens. So you can see the kerogen atoms in gray, asphaltene and resin in purple or violet. And you have the methane, you have the intermediate species. And I would like for you to focus on the space in the center over here. As I play this video, we're going to be producing some of the fluid molecules from within the kerogen into this space over here, from within this kerogen into this space over here. And I ran this simulation. We finally reached equilibrium. And what you can see over here is that there's a whole bunch of methane. There's a whole bunch of methane, some of the intermediate species, some of the propanes and butanes and octanes and things like that, some of them. But we don't see any asphaltenes. We don't see any of the other heavier or aromatic species, perhaps one or two molecules, but that's about it. And so essentially what we're seeing with the simulation is that because some molecules are adsorbed, like this asphaltene molecule, this large asphaltene blob over here, they are less mobile, but the fluids that are free, that are things like methane and ethane, which are relatively free, they are mobilized more easily and are produced more easily. And so here's another picture that I'm going to show you. I'm going to show you the center of mass of butane molecules on the left-hand side. And these red dots represent the center of mass of the butane molecules. On the left-hand side, I have a Kerogen blob. On the right-hand side, I have this Kerogen blob. And here's a 13 nanometer space. And as I run this simulation, we're just focusing on the butane molecules. And you can see that quite a few butane molecules do end up in this space over here and eventually can become producible or are available to be produced. Okay, that's fine. Let's look at the octane molecules on the right-hand side. I have the center of mass of the octane molecules. Again, we're just looking at the center of mass. We're not looking at the entire molecule because it's easier to visualize. And now I'm showing you what's happening with octane. You can see that except for maybe one or two octane molecules that somehow free themselves from the surface and end up in the fracture, most of the octane molecules continue to remain adsorbed within the Kerogen matrix. And what does that mean? It means that just looking at the trajectories of the molecules, comparing what you're seeing on the left versus what you're seeing on the right, it stands to reason that you're going to be producing more of the heavier species than the lighter species, even though your pressures are above your saturation pressure. Just because some fluid molecules are adsorbed and some fluid molecules are free. And again, depending on the affinity between the fluid molecule and the pore surface, you're going to have different species being produced. Okay, so having said that, how can I use this information in any way? So here's where perhaps most of us who are reservoir engineers would be interested, because you are not directly going to be using molecular dynamics, but you'd be using some of the products of molecular dynamics. So one of the things that is an important phenomena for governing transport in these extremely tight shales is diffusion, which is driven by a concentration gradient. And using molecular dynamics, we're able to actually quantify the diffusion coefficients for each one of the hydrocarbon species. And so now you can take this diffusion coefficient, it's on the order of 10 to the negative 10 meters squared per second. You can take these diffusion coefficients and you can input them into a reservoir simulator, irrespective of which reservoir simulator you're going to be using. Each and every reservoir simulator out there does accommodate diffusion. So you can actually model the diffusion of different species in these extremely tight pore spaces. And of course, this is not just a modeling construct. We verified these things experimentally as well. And more importantly, here is what I wanted to show you. We also ran the same experiment with a volatile oil. So this is the volatile oil composition. And because we're only producing the lighter species associated with this volatile oil, what we end up producing is just a gas condensate. This is what we're producing. We're producing a lot of methane, some amount of ethane, some amount of propane, and so on and so forth, but we're not producing any of the heavier species. So here's the question that we need to begin asking ourselves, which is still an unresolved question. If we are sampling at the surface, and this is what we're sampling, what does this tell us about the fluid that we have in situ? What does it tell us about the fluid that we have downhole in the subsurface? Perhaps nothing. And that's one of the things that continues to be a research question. For the most part, People overcome this problem by actually taking preserved core plugs. So you go in and then you take a pressurized core plug and then you bring it up to the surface, and then you actually break up the core and then you extract the fluid, and then you would perhaps have an idea of what you have in the pore spaces. But there are limitations to that as well, because there's only a limit to the size of the sample that you can collect. But this is an important question that molecular dynamics raises. What are we collecting on the surface? What are we studying on the surface? Is it even representative of what's in the ground? And if this is what you're producing, what are you leaving behind? Because if you're talking about shales, typically people talk about 5 to 10% recovery of the original oil in place. And if you're talking about only a 5 to 10% recovery, you're leaving behind 90% of the target. What is the composition of that target? That becomes very, very important. Why is it important? Because if you're going to be injecting a solvent to recover that hydrocarbon, the solvent that you're going to be choosing is, of course, going to be dictated by the pressure and temperature conditions in the reservoir, but more importantly is going to be dictated by what's left behind in the pore spaces. And so knowing what's left behind in the pore spaces, what kind of solvent you need to be using, and things like that becomes really very important. And so that takes us to huff and puff enhanced oil recovery. And EOR has been, of course, worldwide, quite popular, especially solvent injection, or at least miscible gas injection. In unconventional shales, it's something that's become important more recently, over the last, I'd say, 5 to 10 years, just because, as I mentioned earlier, we only recover about 5 to 10% of the original oil in place via primary production, and we're leaving all of the heavier stuff behind. And we're leaving 90% of the oil behind. So it motivates us to begin to start injecting lots of the gas that we're producing back in the ground and trying to recover more of those hydrocarbons. Here is a field experiment that I'm showing you. It's a field experiment of an enhanced oil recovery project. You have cumulative oil on the y-axis, you have time on the x-axis, and you can see the period of primary production, nice smooth monotonic increase. And then, of course, after some time, this plateaus out, because this is when the company started something called huff and puff EOR. And I'll describe huff and puff EOR in a minute. But they shut their wells in for a period of time, and then they brought them back on after injecting a solvent. And you can see that incremental recovery. Then they shut it in again for injection of a solvent, then brought it back on. And you can see that if you were to just take this all the way out over here and extrapolate what you would have done if you were doing primary recovery, we've recovered roughly about 30% more oil. So 30% more oil is a significant amount if you think about the fact that the United States is currently the largest oil producer in the world. So 30% more oil is a lot. Keeping that in mind, typically when we talk about miscible floods or miscible solvent injection, we're talking about a gas injection well on the left-hand side. Perhaps on the right-hand side, you have a production well, and both of them are operating simultaneously. You're injecting the solvent comes into contact with the oil, and it essentially dissolves in the oil or modifies the oil properties in some fashion, and that oil is mobilized towards a producing well and is pumped back up or produced back up. And this is operating in a continuous fashion. In huff and puff recovery, the situation is slightly different. The reason why this continuous injection will not work in shales is because we need to have communication over extremely large distances. And I showed you the shale pores. They're extremely, extremely small. And if you're talking about the permeabilities, you're talking about tens of nanodarcies or at the most hundreds of nanodarcies. So if you were to inject something over here today, it would probably be, you'd probably go out maybe only 3 or 4 inches in about a week or 2. So that's not too far. You want to be able to send your fluids out over extremely long distances if you have the connectivity. Since we don't have the connectivity, what we do is something called huff and puff. Here's an example using carbon dioxide. We have one single well that acts as both the injector and the producer. I inject the solvent into this well. It goes into the formation, comes into contact with the oil, modifies its properties in some way. Of course, we're ensuring that the pressure and temperature conditions are just right. And it modifies the properties of the oil in some fashion. And then we shut in the well, and we allow the solvent to come into contact with the oil, move out, and let the pressure stabilize and do work its magic. And then after maybe about 2 to 3 months, or maybe, sorry, not 2 to 3 months, but maybe after 2 to 3 weeks or a month, we open back the well, and then we produce this CO2 rich oil or solvent rich oil back to the surface. So that's called huff and puff, where we're pumping in, shutting it down, and producing the well. What's the signature of that? It's going to look like this. Here we're looking at flow rate versus time on the x-axis. With primary production, you have continuous production, but then if you're injecting a solvent and then allowing things to soak, that's where you have a shut-in, no oil production. Then you bring the well back on, you have this production, then a very, very rapid decline, because keep in mind, we're not going very far into the formation. With shales, maybe a foot or 2 feet, or maybe a couple of inches, depending on how long you're injecting and the volume of you're injecting. Then again, injection and soaking, then some more production, again, some injection and soaking, some more production, and so on and so forth. So you can see that this is a cyclical injection. It's also called cyclic EOR. It's also called huff and puff EOR. Good. So we want to see what we can recover if we were to inject a solvent, and how would it be recovered. So one of the things that we did for a company was model this process. They were interested in knowing what their produced gas would do. Their produced gas, fortunately, had a C1, C2 ratio of about 70 to 30. And they wanted to see what would happen with that solvent, what hydrocarbon species would be mobilized, and how much of it would be mobilized. So what we did was we again took this Kerogen blob on the left-hand side, Kerogen blob on the right-hand side, and instead of an empty space out over here, think of it as we're putting solvent in there, and then we're going to allow the solvent to migrate into the Kerogen and recover some of the hydrocarbons. Let's see what happens. So here's a video. So you can again see the solvent molecules at C1, C2 mixture. There's a Kerogen blob on the left. There's a Kerogen blob on the right. And I'm just going to play this video. And there's really nothing you can see in this video. You can, of course, see the solvent molecules going out into the Kerogen. And if you were to just stop the simulation out here, you can see that most of the density of the solvent is adjacent to the frac phase, or this is where the solvent goes. Very, very few solvent molecules go really, really deep into the reservoir or deep into the model itself. So, well, there's nothing interesting about that. Let's see what happens in terms of production. So now I'm rendering only the fluid molecules that are going to be produced. The solvent is still in this space over here, but I'm not showing it. So I'm just going to show you what are going to be produced. If you recall previously with primary production, you only had methane and you had some of the ethane fraction perhaps being produced. Now, with exposure to the solvent, let's see what gets produced. You can still see quite a bit of methane being produced, but you also begin to see some of these larger molecules being produced. You also you can see some of these larger blue molecules. You can also see some of these orange molecules being produced. And that's important because now I can run the simulation for an extended period of time and begin to start quantifying what are the hydrocarbon species that are being produced. So if you recall this picture from about a couple of slides ago, where I plotted the trajectory of octane molecules during primary depletion, we saw that hardly any octane molecules were entered this empty space over here to be produced. Most of them were just oscillating in their original positions. That's just thermal oscillation, and you can see them doing that stuff over there. They're not being produced. Very, very few octane molecules are being produced. On the right-hand side, if there is solvent in this space over here, we're going to see the center of mass of the octane molecules during that process. And it would be very, very hard to argue that the solvent is not doing anything. Just by the density of the trajectory that you see in the center out over here, it's very, very clear that you're producing vastly larger number of octane molecules in comparison to primary production. And so this, with molecular dynamics, you can begin to start talking about which hydrocarbon species you're recovering and what kind of solvent you should be using to recover what kind of hydrocarbon. Perhaps a mixture of C1 and C2 is enough to recover some of the octane and some of the slightly intermediate components, but perhaps I still can't recover some of the asphaltene fraction, and maybe we might need something more aggressive. And so that's what we can do with molecular dynamics simulations. Here is tetradecane, for instance. With primary depletion, no tetradecane being produced. But if you introduce a solvent, you can see quite a few of the tetradecane molecules being produced. Dimethyl naphthalene, again, hardly any under primary depletion, and just a few more with under exposure to a solvent. So in other words, what we're seeing is that we can begin to start quantifying what we're going to produce under the influence of a solvent. We can look at methane, ethane, and propane. We can look at the initial oil composition. We can see what was produced under primary recovery and what was produced during soaking or during the enhanced oil recovery process. So we can do those things, and we can begin to start answering questions about what we observe in experimental data. In experimental data, what we do is we take small little pieces of core and which are saturated with the oil, and then of course we expose them to a solvent, and then we produce after exposure to the solvent, we see what hydrocarbons we're producing, and essentially what we produce is lots of the C13s. I'm going to go back one sentence. Under primary recovery, we produce lots of the lighter components, and we know that. I showed you that with molecular dynamics. And we don't produce too much of the heavier components. But when we start exposing the rock to some solvent, we begin to start producing some of the heavier components as well, and increasing larger amounts. And it shows that what you're seeing in experiment is exactly what you're seeing with molecular dynamics. So if you want to explain what you're observing with experimental data, you can begin to start modeling some of that stuff with molecular dynamics. And so it was a very nice marriage between experiment and modeling. The other thing that's you can do with molecular dynamics is begin to start asking questions about how much of time should you inject for, and how much of time should you keep the how much of time should you expose the formation to the solvent? What period of soaking should you be using? Typically in the field, we're talking about 1 month of injection, maybe 2 to 3 weeks of soaking, and then followed by another month of production. And typically you would follow that sequence over and over again. So the question is, why is it 2 or 3 weeks of soaking? And why is it 1 month of injection? Why can't it be 2 months of soaking and then perhaps 1 month of injection? And you can design experiments that would do exactly that. And I think I'll skip through that for right now. But there's lots of agreement between what we did with molecular dynamics and what we did experimentally as well, what really matters is not the specific time of injection or the specific time of how long the rock is exposed to the solvent or the soaking time. What really matters is the soaking and the production time. And what we see is that if you are looking at cumulative recovery as a function of the number of cycles, of course, if you give it a 6-hour soak time experimentally, you would be recovering more oil, and you would think that, hey, we should give it a much larger soak time or expose the rock to the solvent for an extended period of time. Because if you were to use a 3-hour soak cycle, what you see is that it takes 1, 2, 3, 4, 5, 6, 7, 8, 9 cycles to reach about the same recovery as a 6-hour soak cycle. And so you'd begin to say that, hey, a 6-hour soak cycle is more beneficial for us. But if you were to plot this exact same data, plotted as cumulative recovery versus something called residence time. Residence time is your soaking time plus your production time. If you do that, irrespective of whether you do a 1-hour soak or a 3-hour soak, which is shown in red, or a 6-hour soak, which is shown in green, the data all falls on the same trend line. And that's extremely, extremely important, because if you are an operator deciding on whether you should try 2 weeks of soaking and a month of production, or 2 months of soaking and half a month of production, some of this information will allow you to optimize some of those cycles. And we see exactly the same thing with molecular dynamics as well. We can also quantify how deep the solvent goes into the formation and things like that, and scale it up to a much larger scale. I apologize that I'm going a little bit faster because I'd like to end this at the 1-hour mark. But really what we see when we take the molecular dynamics results and scale them up to the reservoir scale or the well scale, the solvent penetration, it does not exceed more than a few centimeters or a few inches. So the reason why increasing the soaking time inordinately does not provide any additional recovery is because the solvent can only go in this much, because the formation is extremely, extremely tight. So just because you increased your soaking time to 3 weeks or 4 weeks is not going to be providing you any additional benefit. So it actually pays if you keep the soak cycles a little bit shorter, allow the solvent to penetrate into the reservoir, but don't let it linger over there doing nothing. So that's an extremely important conclusion. And that's borne out by experiments in the field. Most of the companies who are implementing EOR right now, huff and puff EOR, are of course injecting large volumes of solvent, and they're going through short soak cycles, because a soak cycle is where you're just letting the oil come into contact with the solvent and you're not doing anything. You're not making any money. Where you're making money is the production time. So short soak cycles allows you to start producing more rapidly. And we can see all of these things with molecular dynamics. We can, and also supported by experiment. There are many, many other things that we can do with molecular dynamics. We can model surfactants and surfactant microemulsions, see how we can deliver these microemulsions through frac fluids, and then send them down to your shale while you're fracking, and then see what kinds of interactions these surfactant blobs or micelles have with the surface. Are they able to clean up the surface and recover some of the asphaltene fraction to promote well productivity? These are the kinds of questions that you can begin to answer. Those are the kinds of things that we are working on, and things like that. And so with that, it was a long talk, but I hope that there are a few takeaways from this presentation. One is that if you're going to be talking about really, really small things, there's no other way to understand these processes. You can run some experiments, but then again, the interpretation of experiments can be associated with some ambiguity. But if you have modeling that supports the experiments, and in this specific case, it's molecular dynamics modeling, then of course you would have more confidence in your experimental results. You'd be able to explain your experimental results, especially when we're talking about things that you can't directly observe. And so yes, it's molecular dynamics. It's a fascinating tool. It's unlikely that every one of us is going to be using molecular dynamics, but it's highly likely that several of us would be using results from molecular dynamics. So if you are somebody who does reservoir simulation, for instance, you would be inputting things like diffusion coefficients. You would be modifying the phase description of your hydrocarbon species in your PVD software. You would be doing things like that. And all of those things come from molecular dynamics simulations. And of course, it's not the solution to everything. it comes with its own challenges. So the challenge is that if I want to run if you asked me to run a simulation right now, I would come back to you in December and tell you that here are my results. And that's, of course, an inordinately long time. I can't run it on my laptop. I'd have to send it to the supercomputer, and they would run it over there, because I'm talking about the motion of 500,000 molecules over 10 nanoseconds. It's not a lot. It's not a huge space. It's not a huge period of time. But even that short of a simulation over such a small time scale takes a huge computational effort, because you're talking about the motion of 500,000 molecules and atoms. And I think I said all I had to say in this specific talk. Thank you all for being here this evening, and I'm open to questions.
That's brilliant. Thank you, Deepak, for really a real tour de force looking at molecular dynamics. I've got just 40 years ago, my first degree was in theoretical chemistry. And I remember one of our exam questions was to derive, using Hamiltonian operators, the interaction of two hydrogen atoms. Mm-hmm. And that was about 3 or 4 pages, or maybe longer than that, I can't remember now, just a pure mathematical calculation. Right, and we asked our tutor, said, his name was Balint-Curti, in case that name is familiar to you, Balint-Curti. I said, what about 3 atoms? He said, well, we haven't solved that one yet.
Right. And you're absolutely right, Tim. With 3 atoms, it's not analytically solvable.
So cracking, it was so funny. But yeah, okay. So I've got a stack of questions, but let me pass it to, like, Adrian, you come on. Does that mean you want to ask something? So go ahead, anyone else who wants to ask a question.
Deepak, that was fantastic. I'm just very, very glad that it's finally getting higher profile. So I used it in 2018 on the Boland Shale and the Haynesford Shale. We did a project with the EU, so it's all that stuff's published. So there with University College.
Okay.
Striola.
Oh, very nice. I worked with Striolo. So Striolo was here with me at the University of Oklahoma.
Right. He had 8 students running this stuff. Yep. And so the question is, I mean, it's nanoscale. So it turned out the physics was all different. So is that being understood now more? Because they measured the solubility of methane, and it was 3 times larger at nanoscale than it was in what I call real scale, petroleum engineering scale. So have we understood the physics better at this nanoscale? Because it's not the same stuff.
You're absolutely right, Adrian. And if you do run into Alberto again, please say hi, because we used to work together once upon a time. He was just a couple of floors down from me at the University of Oklahoma. He then moved up to University College London, so I know him quite well. But having said that, you're absolutely right, Adrian, about the physics being completely different. And just to give you an example, we all know that water freezes at 0 degrees centigrade, but when it's confined in really, really small pores, its melting point actually is suppressed. It becomes -2, -3 degrees centigrade. And that has huge implications for what we're doing, say, for instance, in the permafrost. So if you're going to be putting up some structures and things like that, and if you're talking about climate change and other issues, if water is going to start melting at -2 or -3 degrees, or if ice is going to start melting at -2, -3 degrees centigrade, as opposed to 0 degrees centigrade, that has huge implications for all of the structures that we have in the Arctic. So that's something that geophysicists have known for a very long time. We're seeing the same thing as well in these shales. We're seeing something called bubble point suppression. So if you were to take a fluid sample and observe or model it, not model, but experimentally measure its saturation pressure at a specified temperature, it might be like 5,000 psi or something like that. But the same thing, which is confined in a very, very small space, would have a suppressed bubble point, maybe closer to 2,000 or 3,000 psi. And we're seeing that with the dew point fluids as well. And as to your point about the solubility, absolutely. I mean, methane solubility has been shown to go up by close to an order of magnitude, depending on the pressure and temperature conditions.
That's interesting. So carbon dioxide solubility, what about that then? Going back to the obvious thing of sequestration.
That remains a question for me to answer. So, yeah.
Well, see you next year then, shall we?
Yeah. No, so I'm glad that I'm having this conversation. So that's,
They suggested that we need to look at EOR, so I'm glad you've done the EOR bit. In 2021, I'd say exactly what Tim said. Could someone please look at CO2, because the temperature is astronomically negative sometimes. It's very, very cold with CO2. And I'm pretty sure, I know Imperial College say they understand everything about plumes, but I don't think they have a clue, because it's at your scale that we need to actually do some simulation. So I look forward to your talk in 2025, because
Well,
4 fields with CO2 injections, and no one's looked at it. They've never even thought about it. But I have to say, before I leave it on my side, we knew this back in 20, 30 years ago, because the geochemists, there's a group at Newcastle, and they said, what you produce doesn't tell you anything what's actually still down there. And these models finally showed it. So great work, and please come back in 3 years' time, and please explain to us what CO2 actually does in a reservoir.
absolutely, I will. Thank you.
This is great stuff.
There's so many things I want to ask you. So when you were doing that, did you ideally look to see whether or not you could repeat Langmuir's work on the physisorption of those molecules? Can you predict them?
We did not, Tim. Yes, I mean, here's the thing. I mean, we did do some very, very preliminary work on just graphite surfaces, looking at adsorption, but no, we didn't try to replicate any of the older publications or older pieces of work, because what we did was we looked at our own results and then compared them to what we were measuring in the lab, and had some correspondence. We went with it, but no, we didn't go back to Langmuir's 1929 work. But very valid point. Yeah.
Okay. And the other thing is, I mean, Fatai, unfortunately, because he's 4 hours ahead of us, he's gone. But I mean, the obvious question to ask is that you're taking several months to simulate these molecular interactions. Is, well, what can you do with data science to accelerate that process?
And that's a fantastic question. I didn't even touch upon it, but let me just show you. I did put it in my slide, but I didn't talk about it. So when I was talking about this protein folding problem over here, this protein folding problem was solved for the first time in. Can you see my screen, all of you?
No, it's me. I've just got to move across a different screen. That's why I was looking in the wrong direction.
Okay, yeah. So if you look at this protein, if you look at this protein over here, this protein structure, these two protein structures were solved in December 2020, not too long ago. And these were actually solved using molecular dynamics, accelerated with machine learning. So, which is fascinating. I mean, this is a 50, 60-year-old problem, but only now were we able to get reasonable accuracies in terms of how these proteins come together or fold. And that's just because we were able to use machine learning to speed up some of these simulations. Okay. And talk about some of these larger models.
X-ray crystallography to get to ascertain the quaternary structure. Is that what you would? How do you? Yeah. So you're saying I've got the quaternary structure from the X-ray crystallography. Okay. Now let's try and sim. Oh, very good.
Yeah. So it's fascinating. Yeah. So and this is something that I'm looking into, but for instance, if you're talking about asphaltene aggregation and things like that, or even some of the things that you folks who work with oil field chemicals and things like that, perhaps there are applications out over there that I don't know about, but I think it's an exciting area. Lots of applications potentially out over there. So
hugely, yeah. Okay, and just on the guts of this thing. So, are you solving the Hamiltonians for all these atoms and molecules? So what is the process of the simulation study you're doing? I mean,
okay, so the exact simulation is really exactly what I mentioned to you. So, if I'm looking at this red molecule over here, I need to know what is that red molecule and what kind of a force field it's going to be exerting to what distance out. So, we express that as a Leonard-Jones potential. And so then, of course, that has a radius, the Lennard-Jones potential, and then you have some atoms within that radius, and each one of those atoms is going to be affected by that specific Lennard-Jones potential, and it's going to move in a certain direction. But at the same time, this atom is under the influence of other forces as well. We're essentially just solving Newton's laws of motion. Force is equal to mass times acceleration. That's it.
Interesting. Okay. Okay. So you're not trying to not trying to invoke any particular quantum effects or anything like that. The way I was thinking of it with Hamiltonian. So you're not doing it that way. You're simply saying, I've got point forces.
Yeah.
That's interesting.
So
Okay.
going to your point, I mean, the thing is, there are people. So where do I get the force fields from? How do I know that hydrogen behaves this way? Or how do I know that nitrogen behaves this way, or oxygen behaves this way? Fortunately, there are people who've done what you're exactly asking about.
Yeah.
They are taking the protons, they're taking the neutrons, they're taking the electrons in their cloud and modeling the behavior of that atom. And telling us what kinds of force fields this specific atom would be exerting.
And then you're simplifying that by saying, okay, well, roughly speaking, this will cover what you've just told me, and then you simulate it more easily. Otherwise, you'll be an impossible task to compute it.
Right.
And you know, when you put that Kerogen molecule up, so how did you predict the folding of that molecule? Later on in the presentation, you showed it folded itself. Yeah, or they're about 6 there, aren't they, or something? Or 16 there.
16, yeah. So how exactly do we know that this is sort of an equilibrium configuration and not any other? So we start with pretty much the same random placement of all of these molecules and atoms in over there, and then we go through that simulated annealing process, and we end up with maybe 10 different configurations, and then we can measure the potential energy associated with this configuration. And there are going to be some of them associated with the minima. And of course, it may not be a global minima, but at least if you reach a local minima, then you're happy with it. So we don't work with just one model. So I'm just showing you one model, but in this whole sequence of simulations, we actually use 3 different models.
Sorry, Adrian, I spoke over you.
So Van der Waals interaction. So how are you making sure that's correct? Because this is where I thought where maybe the models were going wrong.
Could you repeat that, Adrian? Sorry, I didn't catch you.
Van der Waals interactions of Van der Waals.
Yeah.
How are they being replicated in your modeling? Because this is where I thought the only Achilles heel was at the University College stuff.
Okay, so you're right. Okay, so I know the Van der Waals interactions for a carbon atom very, very well. I know the Van der Waals interactions for a hydrogen molecule or a hydrogen atom very well. Here is where we're going to be making a leap of faith. If I'm going to be talking about a molecule that's perhaps this big, I'm saying that the interactions of this huge Kerogen monomer is the superposition of the interactions of each one of these individual atoms contained within the monomer. And people have shown that it's valid as long as you don't go to inordinately large molecules or large structures. So for something that's about this size, people have shown that it's fine, but we didn't prove that that's necessarily the case. We could very well be wrong by a certain amount, no doubt about it.
In our reservoir simulation models, we always have tuning things. So first of all, this is a problem with reservoir simulation. Well, 3 problems with reservoir simulation. Firstly, we don't understand permeability, or most people don't. And the second thing is the tuning, and then obviously it's the time. So your simulation takes 3 months or 2 months. How do you know there isn't some sort of mathematical error that's creeping in, doing all these computations? It's a hell of a lot of computations. So how do you know that the computations are actually correct? But you know, you're obviously getting precision, but are you getting accuracy? So let's put it that way.
You're absolutely right. So the only way that you can ascertain that perhaps nothing's going wrong is ensure that when you're done with your simulations, you have reached what we call as equilibrium. So the pressure looks like it's flattened out, the temperature looks like it's flattened out, the energy levels look like they're flattened out. Now, of course, we can start with the initial configuration that would bias you towards the correct solution or what you think might be the correct solution. And so when you're running these simulations, the reason why it takes such a long time is because we start with multiple initial configurations, random placement of molecules, and we let each one of these initial configurations evolve in time. And they should all reach the same solution. If they all reach the same solution, we say that, hey, this is a reasonable solution. I can believe these results. Of course, the more the better, but there's a limit to how many we can run. Typically, again, if you speak to Alberto, he would run about 3 to 4 different simulation runs. I run about 3 to 4 different simulation runs.
It's definitely it's a very exciting tool. So I do think CO2. I'm not too sure why people haven't got into it yet, but it is a hot topic. I know in the UK we don't have any CO2. We're running out of it, so we don't have enough to put in packaging and stuff like this and food and stop fertilizer production. So even though the planet's warming up with CO2, we've got no CO2 anywhere else. So maybe people should take CO2. But anyhow, really good, Deepak. Thanks very much.
Thank you very much.
Have you tried to mimic any of the statistical thermodynamic calculations, Maxwell, Boltzmann, and that sort of stuff, to see whether or not you can effectively demonstrate entropy in action and make thermodynamic predictions?
Yes, we actually did something like that. I don't have them out over here. This was like a long time ago, just when I was getting started, when Alberto and I were working together. This was like in 2014. I'd have to go back and look at some of the things that we did to answer your question. But yes, but since then we haven't done any of that stuff. So.
Okay, it was my research project, but of course I didn't have any of these tools. It was all we did pencil and paper, but trying to demonstrate the same thing using alkanes, a couple of alkanes, dodecane.
Yeah.
I mean, it's a real trip down memory lane to see what's happened in 40 years since I was involved in this. So it's utterly brilliant. I can't say how much you've made my evening. It's just.
Thank you very much. That means a lot to me.
Oh, well, okay. So those of you who are not chemistry geeks, anyone else, do you want to say anything, or anyone else want to make any comments or ask any questions?
A quick question from me. So in the tight reservoir, it seems that what we take as a bottom hole sample, and me with my kind of limited production engineering mindset would probably put into every single simulator as the representative of the fluid, might not actually be the fluid of the reservoir. Could similar problems be present in non-higher Darcy sands or non-tight reservoirs? That the production, what we would take at the bottom hole, would not be representative of the reservoir itself.
I've thought about that question, Adam, and unfortunately, I don't know. My reading may be limited, but I think the literature is a little bit silent on that. But I would imagine that if you had an oil wet reservoir of some sorts, you perhaps may not be collecting representative samples of what's in the subsurface. But that would be my only guess. Beyond that, yeah, my guess is as good as yours. So.
Does anyone else want to ask any questions, have any comments?
Well, we'll come to that. As I said, Newcastle did a lot of this about 10, 20 years ago. Mm-hmm. So, I mean, they're just the trace. So basically they reckon that you could trace fluids to particular wells. And it doesn't change the PVT, but it's like a fingerprint. So that's what their view was. That's obviously in Darcy sands, or it's not. But even this stuff, I mean, you're just talking about one nanometer, so it's not that different. So, yeah. Although the physics is slightly strange, but yeah. So we have done the right thing. We just didn't realise that we maybe weren't as accurate as we should be. But we've been accurate enough. It doesn't mean we're going to re-sample every single flow rate for every single well in the whole of the North Sea. I mean, we're bang on. It's back to measurement. Measurement doesn't give you the answers, which is why everyone says you got to measure something before you can manage it. That's rubbish. It's the stuff we're not measuring is the stuff we need to measure, and that's climate change. So we're not measuring the right stuff, right, and we're not actually subscribing it. But what we measure in the field, it's near enough. But the fingerprint, there is a fingerprint. That's what these geochemists talk to. Geochemists.
Any other remarks? Well, Deepak, I think we're saying goodbye to you. Thank you so much for an absolutely fabulous, absolutely brilliant. And if you're able to share a sanitized version of your presentation, then